speaker, who is doctoral researcher Tuomas Borman from University of Turku, and he is the key developer of the microbiome data analysis framework MIA. I want to say that Tuomas and his colleagues are doing extremely important work in developing data analysis infrastructures for microbiology researchers, which include open source code, expert guidance, training, and also dissemination of the best practices here. So it's really great to have you here today. And he will, I think he will tell us also more broadly about the Bioconductor bioinformatics project, which Johanna also mentioned, and especially the MIA ecosystem. So please, Tuomas.
Yes, modest. Share my screen now. Now you should see my screen like this. All right, thanks for nice words. Yes, this is Tuomas calling, and in this presentation, or this half an hour, we are going to discuss about first half is this Bioconductor, and then we are going to discuss about this MIA, which is the microbiome analysis framework within this Bioconductor. It was already mentioned in a previous presentation, so Johanna, fasten your seatbelts. We are ready to take off, as they say. All right, so first we are going to discuss about Bioconductor. What is a Bioconductor? So you might be already seen this logo, but it is like it comes like conducting, you know, the music conductor, the thing that like keeps on swinging hands. So it comes from like conducting biological data analysis, something like that. And you might, if you know Bioconductor, what it is, then you might think that it's like Google Play or Apple Store of bioinformatics, but it is actually much more than that. So I really want to emphasize this. So it is not just a platform for software, but much more. And someone has said that it's world's largest bioinformatics project, but it is kind of hard to measure. But it might highlight the impact of Bioconductor. So what it is? So it is Bioconductor is worldwide community of bioinformaticians. I would put it like that. So it is a community of people, and it is there's in this community there are people that are software developers, but also software users, because there's a unique set of talents in both worlds, which are required to solve these complex biological problems. And what Bioconductor does, it puts these people on the same table to discuss about these problems. And that is why there's different activities going on in Bioconductor. There's this training and workshops. So that's like because the aim is to train and advance this bioinformatics field in general, train to those current and future bioinformaticians, and also to facilitate networking and the collaboration. There's these conferences and community forums to discuss about this bioinformatics. And of course, lastly, but not least, it is the bioinformatics software. So about this bioinformatics software next. So currently there is over 2,000 different R packages in Bioconductor, and there's also some activity in Python, but the R is the main language. And when package or software is submitted to Bioconductor, it went through this review process. So there's automatic review also, but some person from Bioconductor will check that the software fulfills the Bioconductor requirements. And these are related to documentation, but also the code. And if it does not fulfill these requirements, it will not be there in Bioconductor. And that's not enough. Also, there's a weekly testing. I think several times in a week, those packages are tested that they work as intended. And if they are not working, then bye-bye. They are removed from Bioconductor. And this makes also Bioconductor unique, because there's not, for example, in CRAN or Python package index, there's not this kind of strict instructions and rules, and that makes the Bioconductor software reliable, and also facilitates or encourage developers to keep on developing those software. And in this figure, you can see that there's this number of packages from 2009, I guess, to this date. And you can see that the number of packages has grown a lot. And there's this genomics is the largest number of packages, and microarray, and then there's single cell has been grown a lot in past years. And here is the microbiome field. It is still relatively small, but it's expanding, and there's currently around 50 microbiome packages there. Then this software development in Bioconductor, it is based on these data containers. I will shortly tell you what are those data containers, but they are structured way to present and store the data. And then around these data containers, developers can develop those methods and software, because they can rely that the data is in this structured format. And for user perspective, it means that when you have the data, you put the data in this data container, then you can easily apply methods and manage the data. So that makes the life much easier when you put the data in this mystic data containers. And of course, there's this community that surrounds the whole thing, because that's the essence or the main thing about Bioconductor. But what are those data containers? Hopefully your office does not look like this, but it's these are generated with AI, so that's why they look a little bit stupid. But anyways, this on the left side, this illustrates a pile of pages. So it's a complete mess, and it is a mess. You don't know where's which page and where is what information, and it's very hard to find anything from there. But then on the right side, there's a book. For the younger people, it's like this kind of organized way to present pages and stuff. But that makes it like this is highly optimized for sharing text or pages, this kind of book. So I would now ask you, you don't have to answer for that, but would you rather read 300 pages from this, like of course you have to find those pages first of all, but would you rather read 300 pages from this mess of pages, like this pile of pages, or would you rather read 300 pages from this very nicely organized thing called book? Yes, that is stupid, silly question. Yes, of course you choose the book, because that's the very easy. You know that after page 5 you get page 6 and so on. From the table of contents you can find which kind of information you are searching for and so on. And that is actually my main point of this presentation. If you only remember one thing, that should be put it in a book. That's how it goes. Very simple. Put it in a book. Because the same thing applies to data. So when you have this microbiome data, or usually this biological data, it's so complex that you cannot present that in one single table. So you have multiple tables. So for instance, after you have mapped those sequences, you have the abundance tables. Then what you have is the taxonomy table. Then you have the sample metadata, and so on and so on. So instead of analyzing the data as a pile of mess, like pages, like you saw that picture on the previous slide, you put it in a book. So in this case, in the data context, it is the data container. So now you know what is a data container. What is the idea of those? So yes. So in Bioconductor, as I mentioned, the data containers are the core structure. And this summarized experiment, it is the most widely used data container in Bioconductor. And it is optimized for biological data. It is extended to different purposes, which means that for instance, this summarized experiment we have because let's take an example, microbiome field. We are talking about that in this webinar. So in microbiome field we have those special needs that are not fulfilled with this plain summarized experiment, but this is extended this summarized experiment so that those requirements are fulfilled. But they are still summarized experiment, or inherits the same structure, which means that we can cross-utilize those methods. So that's that is important to note, that they are still summarized experiment, and that's why we are talking about those now. So here on the pie chart you can see that there's these are the fields, or the number of packages in each field that are utilizing this summarized experiment data container. And you can see here this genomics, and then again microarray and a single cell. And here is the microbiome 6 different packages utilizing this summarized experiment data container. So let's talk about this summarized experiment a little further. So this is the structure of summarized experiment data container. Here, here are the abundance tables on the center of this figure, and that actually creates the core also of this summarized experiment data container. And here, this can be, for instance, counts or other gene or other microbial abundances or something like that. Doesn't matter. Numeric matrix. And we can have here multiple different abundance tables. For instance, if we do transformation, it is makes sense to put it in the same data structure because it's represent the same data. So we can have multiple abundance tables here. And here the rows represent microbes. And then columns represent samples. And for both columns and rows, we can have metadata. And here the column metadata or sample metadata, it represent the sample metadata. So the sample type or description of samples, and here we can have row metadata, which is the taxonomy table. So in which taxonomy group this certain microbe belongs to. And then we can have metadata for the whole experiment, for instance, who took those samples and who did what. So this is the summarized experiment data container, and is widely used in Bioconductor, very well suitable for biological data, and that's why it is used in genomics, transcriptomics, microbiomics, and metabolomics, proteomics, and so on. But what is optimal data container for microbiome data? As I already mentioned, there's different. This summarized experiment is almost optimal, but we need other functionality that is not fulfilled yet with this summarized experiment. So let's list those. Multiple essays. This means that we have this, because usually we have to apply transformations to this microbiome data. So as I mentioned, it makes sense to put it in the same data container. Then hierarchical data. So those microbes genetic relations or relations in general, they are usually represented with these phylogenetic trees. So we have to incorporate that kind of data also to our data container. And this is site information. This is also a little bit related to the hierarchical data point, but there are specific requirements in microbiome field that needs to be fulfilled, and this site information notes that. Optimized. So it is not too uncommon that we have like 20,000 different microbes in our data and thousands or tens of thousands samples if we are analyzing some kind of population cohort. So it needs to be fast and efficient. Integrated. This means that in microbiome studies, it is more and more common that we incorporate different measurements. For instance, metabolomics. That is because metagenomics, they only tell us who they are and what they could do. But to know what they are really doing, we need more data. And for instance, metabolomics. And And this integration part means that it should be easy to integrate this kind of data to the same objects and manage the data easily. That is to enable efficient multiomics data analysis workflows. And what this all means is that this is to reduce overlapping efforts. We can collaborate with other fields and use the same methods for metagenomics or metabolomics data, and also to ensure that this will be also fulfilling the requirements of the field in the future. So that makes this, if we add this 3 to this summarized experiment, that makes it 3 summarized experiment. And it is extension to this summarized experiment data container. And if the summarized experiment were optimal for biological data, this is optimal for microbiome data. And what it does is that it links this microbiome field to larger summarized experiment family of tools. Now you should have like, I have seen this, the deja vu moment, because this is the summarized experiment data container. So you have here abundance tables, here is the sample metadata, here is the taxonomy table, here is the general metadata, and you remember that these columns represent samples, and then these rows represent microbes. And how to get 3 summarized experiment with the easy wrist movement, bada bim, bada boom, it's here. And you can see that there's additional slots. We call them slots, these different like places for data types. So here's 3 data and this column link and row link. But this is still this summarized experiment data container, because you can see here is the abundance tables, here is the sample metadata, here is the row metadata, so the taxonomy table, and samples represent these columns, and rows represent these microbes. But these additional slots are for trees. So here we have this row tree. And what is a row tree? It is a tree for rows, tree for microbes, phylogenetic tree. So here we can incorporate the phylogenetic tree also to the summarized experiment data container. And for that, to incorporate the data to this rows, we have this row link that just creates a linkage between this node and these 2 microbes. And here we have this tip of this tree, which is creates a linkage with this row. And also additionally, this is not too widely used yet, but we can have also 3 for columns. So that can represent, for instance, if we have like samples from father and grandfather or grandmother and from children. So this kind of hierarchy we can also illustrate with this kind of hierarchy trees. And here again, this node here is has linkage with this column right there. So around this 3 summarized experiment data container, which was the most optimal data container for microbiome analysis, we developed this microbiome analysis framework. So it is not only a single R package, but it is several R packages, and this MIA package, it is the most widely used. That is because it contains those common operations in microbiome data analysis, including transformations or agglomeration, data wrangling, and some like alpha diversity and that kind of things. And it is currently top 7, around top 7. I will round this a little bit. Top 7% from Bioconductor downloads. It's quite okay if you remember how small the microbiome field was in Bioconductor. And of course, we have not done everything. This is community driven. There's here is just a list of those packages. There's for common visualization for microbiome stuff. We have this MIA-Viz, and then there's simulation stuff, time series stuff also. But if you're a beginner, you might want to check this MIA-DAS and IC3, which are the provides graphical user interface. So you don't have to be able to code. You can just use those quite easily. And as I said, these are different tools available, and it is expanded further by independent developers. For instance, Netcomi for network analysis and Benchdumming for differential abundance analysis. So why you should use this MIA framework? It has this shared data container called re-summarized experiment or summarized experiment. And why it is a good thing? It is a good thing because it is more and more common that there's in microbiome research there's multiomics data. So we integrate different data sources, and this means that we can more easily integrate the data if we are using the same method, the same data container. That makes it very efficient to develop these multiomics workflows. Compared to phyloseq, for instance, which is also common, it does not have a shared data container, so it is not optimal for multiomics data analysis, which is very common. But of course, phyloseq is much larger, much more mature, so it's the point goes from that to phyloseq. But other advantages of MIA is that it's scalable and optimized for bigger data sets, and it has this very comprehensive documentation. And what this all does is that it allows us to develop this microbiome, also multiomics, microbiome multiomics data workflows. About this documentation, we have this Orchestrating Microbiome Analysis with Bioconductor online book. It is good to mention because in today's world, because of this inflation and stuff, this is free to use. Yes. It is freely available, and it has those resources. We are trying to gather the best practices of the field there. Of course, this means that this community has to be involved. So we are welcoming every contribution there. We can discuss what are the best practices there, and that makes it more like community effort and truly makes it like to gather best practices. And it has also those practical examples how to conduct the research, how to do those, like calculate alpha diversity and stuff. And we can have a quick look. I can see that my time is soon running out. So here is the book. This is the book. We can check, for instance, data context. Here is different topics, introduction and stuff. We can go to data containers, and here we can see how here's the same figure. And we can see here that how the data container tree-summarized experiment looks like in R console. So here we have these different slots. There's essays, which has the abundance tables. Then we have row data for taxonomy table, column data for sample metadata, and so on. There's row tree for phylogeny and so forth. So that is how it looks. Then we can see, for instance, there's this diversity stuff. We have here lots of information how to calculate alpha diversity. Add alpha is the thing. Here's plot call data function. This is good example that we are utilizing here single cell analysis package. So not our own package, but we are borrowing the methods, which is quite nice. And here is the stuff for differential abundance. One big point is that we have here nice references. For instance, from Juho Pelto et al. Nice reference. They compared those methods. And then multiomics we have here. This is expanding. So hopefully this year we are able to expand this. And we have lots of nice ideas for that. And lastly, the support. You can find information on how to contact us. And of course, you can send an email or something also. I would love to show you more, but it seems that it's time is a little bit running out and so on. But you can do that by yourself. And I would like to conclude this presentation with poem. And it goes like this. Put it in a book, make it clear, MIA frameworks waiting here. Oma's guide will light the way, helping you every step of the day. Thank you very much.
Thank you, Tuomas, also for these encouraging poetic words in the end. Now we do have a little bit of time for questions to Tuomas, and maybe also if there were some pressing questions to Johanna still. I have been personally happy to recently be using both MIA and Phylosec, and I can say that this book is really excellent. It's a really nice resource for my kind of let's say wannabe microbiome analysts who don't have that deep expertise in complex statistics. It's really worth checking out. But yeah, I was informed that it might be impossible to send public messages to chat, but can you see your hands? Not sure. So you can also just raise your voice or send a chat message to me if you can't do it for the public chat. Yeah, just feel free to do that if I don't notice your hand or comment. I think it was nice to hear also, but I haven't tried these graphical user interfaces that you mentioned, but I think that's probably very nice also for many users, considering what Johanna also told about the importance of the kind of visually evaluating the data before you dive into the deeper analysis. So it's probably makes it more easy for many of us to do using these interfaces.
Yeah, they are actually very new, but the idea is also that it makes easy to export the data before the going deeper. So that's a good point.
Yeah, that's very nice, and yeah, and it's also very nice to see that the framework is all the time actively developing. Every time that I go to look at the book, there's something new, and of course these multiomics developments are really interesting and nice to see where it goes.
Yeah, there's methods available more and more, and we are also trying to to improve the OMAS chapters for that, but unfortunately there's too little time, but trying our best.
Yeah.
Can I have one short question, Tuomas?
Yes, of course.
I'm just interested in why you have built the data containers in the way that you have samples in columns and species in rows, because classically in data analysis, data tables are built in opposite ways, that you have samples in rows and
Yeah, I know that. That's a very good question. But actually, this summarized experiment that I already or I introduced, and this 3 summarized experiment, as I told you, it is based on this summarized experiment. So this summarized experiment is very established, and it was established in 2015, I guess. And we have just extended that and created a framework. So it was not our choice, but that's how it goes. It doesn't really make that much of difference from the point of data analysis, but it's just good to note that there are other way in that sense. But good question.
I don't know if one very simplistic practical reason could be that often in this type of data there are lots of features, maybe more than samples. So if you, I don't know, I mean, it doesn't of course matter for the like deeper data analysis, but then you're viewing the table, then maybe it's easier to handle.
Yeah,
this way.
Yeah,
at least sometime.
Yeah, good point.
Okay, if there are no other comments or questions, then let's thank once more Tuomas. It was very nice to have a representative of the MIA core team